Okay, let's unpack this. Have you ever looked at a tiny moss or a towering redwood and truly wondered about its place in the grand scheme of life? Or perhaps thought about the invisible forces shaping our world, from the smallest bacteria to the most intricate flower? Today we're embarking on a truly expansive deep dive into the incredible diversity of life. Our mission is to extract the most important nuggets of knowledge from a stack of sources, specifically Section 4, Diversity from Raven Biology of Plants, the 8th edition. We'll cover how plants and other vital organisms are classified, how they function, and their profound connections to our planet and even, well, our very existence. Think of it as a supercharged shortcut to being well-informed about the vast, beautiful, and sometimes bewildering world of biological diversity.
What's truly fascinating here is that we're not just memorizing names, we're tracing the evolutionary story of life on Earth itself. From the fundamental rules scientists use to organize millions of species, to the hidden lives of microorganisms, and then to the evolutionary triumphs of plants that colonized land. We'll see how scientists piece together this intricate puzzle. We'll explore the why behind these classifications and the how these diverse life forms sustain themselves and interact, giving you a comprehensive yet digestible understanding of this foundational area of biology.
So where do we even begin to make sense of the millions of species out there? It all comes down to systematics and taxonomy, the science of identifying, naming, and classifying species. It's like creating a massive, organized library for all living things. Right. And the first rule in this biological library is the binomial system.
Right, the two-name system. Every species name has two parts: the genus name or generic name, and then the specific epithet. Take catnip, for example. It's Nepeta cataria. Nepeta is the genus. You can use a genus name alone to refer to a whole group, like envisioning 3 different violet species side by side, all part of the genus Viola.
Ah, okay. So viola tells you it's a violet. But that second part, cataria for catnip, or maybe biennis for some other plant, that second name doesn't mean much by itself, does it?
Exactly. A specific epithet like biennis is meaningless alone. It needs its genus. So you could have Artemisia biennis, which is a type of wormwood, or Lactuca biennis, a wild lettuce, or Inothera biennis, evening primrose. They all share the biennis epithet but are completely different plants in different families. You always need both parts, and typically written in italics or underlined.
Got it. 2 parts for clarity. Does it ever get more specific than that?
Sometimes, yes. Species can have further subdivisions, like subspecies or varieties. Members of a subspecies or variety share unique features not found in others of that species. So you might see a 3-part name, like Prunus persica var, Persica for a peach tree versus Prunus persica var, Nectarina for nectarine. That repeated persica in the first name tells you it includes the type specimen for the species, sort of the original reference point.
So why this whole hierarchical naming system? Is it just tidiness?
Well, tidiness helps, but it's much more fundamental. It's about establishing a universal language. It allows scientists anywhere in the world to unequivocally identify organisms and communicate about them without confusion. Before Linnaeus standardized this, local names varied wildly. Imagine the chaos in scientific study. Trying to discuss a plant when every village called it something different. This system brings real precision.
Okay, makes sense. So from individual species, we zoom out to larger groups, like organizing books in our library, maybe first by author, then genre, then bigger literary movements. And you said this categorization has changed over time.
Oh, significantly. Linnaeus started with just 3 kingdoms: plant, animal, and mineral. Kingdom was the biggest category. Later, scientists added more levels. Genera were grouped into families, families into orders, orders into classes. A botanist named de Candolle even added divisions specifically for plants. And a more modern change, phylum, is now used instead of division in botany, matching what zoologists used for a long time.
So the current hierarchy goes species, genus, family, order, class, phylum, kingdom, and domain at the very top.
That's the standard sequence, yes, from most specific to most inclusive. And even that structure is still evolving, especially at the higher levels.
Right, you mentioned that earlier, like with the protists.
Exactly. Until fairly recently, the domain Eukarya, that's organisms with complex cells like us and plants, was thought to have 4 kingdoms: Protista, Fungi, Animalia, and Plantae. But when scientists started comparing DNA, the genetic blueprints, they realized Protista wasn't monophyletic.
Meaning they don't all come from one single common ancestor, excluding other groups.
Precisely. It was kind of a catch-all group for eukaryotes that weren't plants, animals, or fungi. So now, researchers hypothesize that eukaryotes actually comprise 7 major supergroups. These sit between domain and kingdom. All these supergroups include various protists, but fungi, animalia, and plantae still hold their own as distinct kingdoms within this newer framework. It's a much more accurate reflection of evolutionary history.
Wow. So this shift from Linnaeus's idea of fixed categories to Darwin's evolutionary history, that's a huge change in thinking, isn't it?
It's profound. It means our classifications aren't just arbitrary boxes anymore. They're intended to be natural classifications, truly reflecting the evolutionary relationships of phylogeny among organisms. We try to show these relationships using phylogenetic trees, which are basically hypotheses about how different life forms are related genealogically. It's about mapping the actual tree of life.
That's fascinating. But okay, how do scientists figure out those relationships? How do they know if two similar features, say spines on different plants, are because they share an ancestor, or if they just evolved similarly because, I don't know, they both live in deserts?
That's the classic puzzle of homology versus analogy. Homologous structures have a similar evolutionary background, even if their current function is different. Think of the bones in a human arm, a bat wing, and a whale flipper. Different uses, but the underlying bone pattern points to a shared ancestor.
Okay, shared ancestry. What's the opposite?
That's analogous structures. They might have a similar function and even look alike, but they have entirely different evolutionary origins. They're a result of convergent evolution. The classic example is the wing of a bird and the wing of an insect. Both are for flight, but they developed completely independently.
Does that happen in plants too? Convergent evolution?
Absolutely, and sometimes in really striking ways. A great plant example from our sources is a cactus spine versus a hawthorn thorn. A cactus spine is actually a modified leaf. A hawthorn thorn is a modified stem. They both offer protection, look sharp, but they originate from different plant parts entirely. They're analogous, not homologous.
So similar problems, similar solutions, even from different starting points.
Exactly, and desert plants are a prime example of this convergence. Think about it. Unrelated plants in similar dry habitats, maybe even on different continents, often end up looking remarkably alike. They develop features like fleshy, columnar stems to store water, protective spines instead of leaves, and reduced leaf surface area to minimize water loss.
You mentioned 3 different families earlier: cacti, spurges, and milkweeds.
That's right. The cactus family, Cactaceae, the spurge family, Euphorbiaceae, and the milkweed family, Apocynaceae. All 3 are fundamentally different flowering plant families. Yet each includes members that independently evolve these classic cactus-like features. The cactus-like spurges and milkweeds actually evolved from leafy ancestors that looked very different.
And they fill the same niche in different places.
Pretty much. Native cacti are almost exclusively found in the Americas, the New World, but you find these fleshy, spiny spurges and milkweeds predominantly in Asian and African deserts, essentially filling a similar ecological role. Even their internal chemistry can converge. A special type of photosynthesis called CAM, which helps save water, also evolved independently in these different groups. It's really quite amazing.
So how do scientists sort through all this, the homology and the analogy, to build those phylogenetic trees?
One major approach is called cladistics. It focuses on identifying shared derived characteristics features that evolved in a common ancestor and were passed down to its descendants. Cladistics aims to build phylogenetic trees, or cladograms, based on these shared innovations. And a guiding principle is parsimony.
Parsimony, like keeping it simple.
Essentially, yes. The principle of parsimony suggests that the simplest explanation, the cladogram that requires the fewest evolutionary changes, the fewest independent origins of traits or losses of traits, is the one we prefer. It's the hypothesis that explains the observed data with the fewest assumptions of analogy or convergence.
Okay, that makes sense. But you mentioned earlier that things really leaped forward with molecular data, looking beyond just physical form.
Oh, absolutely. Molecular systematics revolutionized the field. Imagine being able to compare organisms based on their actual genetic blueprint, their DNA or RNA sequences. This provides a whole new layer of information, often much more detailed than morphology alone.
How does that work better than just comparing, say, leaf shapes?
Well, for one thing, genetic data is easier to quantify. You can directly count the differences in DNA sequences between organisms. Plus, there are potentially vastly more characters individual points of comparison in a gene sequence than in physical structures. And crucially, it allows us to compare organisms that look incredibly different physically. You can compare a fungus and an animal at the gene level in ways you just couldn't morphologically.
So differences in genes tell you how long ago they split off from each other.
That's the idea, especially with certain types of DNA sequences. Differences accumulate over time, largely due to neutral mutations, at a relatively constant rate in some genes. So fewer differences between the sequences of two species generally mean they shared a common ancestor more recently. More differences imply a more ancient split. Non-coding DNA regions, which don't directly make proteins, are also useful as neutral markers for these comparisons.
Is there a specific gene that's particularly useful for plants?
Yes, plant systematists have focused a lot on a chloroplast gene called RBCL. It codes for a key part of the enzyme ribisco, which is essential for photosynthesis. This gene is great because it's present in virtually all photosynthetic eukaryotes and even cyanobacteria. It also evolves relatively slowly, making it good for comparing major plant groups over long evolutionary timescales. It's a single copy, doesn't have interruptions called introns, and it's long enough to provide lots of data.
Slow evolution is good for big picture stuff, but what about closely related species?
Right, RBCL might be too slow for that. For finer scale relationships, like within a genus or between closely related genera, scientists often use other chloroplast genes, like ATPB, sometimes in combination with RBCL. Mitochondrial genes tend to evolve too slowly in plants for species-level comparisons, but some nuclear genes are becoming more widely used too.
It sounds like a genetic toolkit for building the tree of life. Is there anything like a quick identifier, like a barcode?
Funny you should mention that. There's a concept called DNA barcoding, proposed by Paul Hebert. The idea is to analyze a small, standardized piece of DNA to rapidly identify any species. Think of it like a universal product code for life. For animals, a mitochondrial gene called COX-1 or CO1 works really well. It varies a lot between species, but very little within individuals of the same species.
So you could potentially identify any animal from just a tiny sample using that one gene. That's powerful. Does it work for plants too?
The concept is definitely being applied to plants, though finding a single, universally perfect barcode gene for all plants has been a bit more challenging than for animals. But yes, the potential is huge for rapid identification, tracking biodiversity, verifying ingredients, all sorts of things. Overall, this ability to compare organisms at their most fundamental genetic level has truly redefined our understanding of biological diversity. It's led to much more accurate classification schemes, challenging old assumptions based purely on looks and giving us deeper insights into the evolutionary tapestry of life. For instance, realizing that bacteria and archaea were fundamentally distinct domains, or untangling the complex origins of eukaryotes through endosymbiosis, those breakthroughs were largely driven by molecular data.
You mentioned endosymbiosis. That's the idea of cells living inside other cells, right? Yeah. How did that lead to complex cells like ours?
It's one of the most mind-blowing concepts in evolution, really. The theory is that proto-eukaryotic cells, early, simpler eukaryotes, engulfed certain bacteria, specifically bacteria that were ancestors of mitochondria, the powerhouses of the cell, and chloroplasts, the sites of photosynthesis. But instead of digesting them, the host cell formed a symbiotic relationship, literally living together.
Like having a permanent house guest who pays rent with energy or food.
Kind of. We even see potential analogs today. There's a green protozoan, Vorticella, that houses endosymbiotic chlorella algae. The algae photosynthesize and provide food to Vorticella, while Vorticella gives the algae protection and nutrients. Over vast evolutionary time, these engulfed endosymbionts became much more integrated. They lost unnecessary structures like their own cell walls. Much of their DNA got transferred to the host cell's nucleus, but they retained some of their own genetic material and the ability to replicate themselves. That's why modern mitochondria and chloroplasts still have their own small genomes and resemble prokaryotes. In some ways, they are descendants of those ancient engulfed bacteria.
Wow. And you said this happened multiple times, like layers of engulfment.
Yes, it gets even more complex. Primary endosymbiosis is that initial event. A free-living cyanobacterium gets engulfed by a eukaryote and becomes a chloroplast. This happened in the ancestor of green algae, red algae, and land plants. But then came secondary endosymbiosis. This is where a eukaryotic cell that already has a chloroplast from primary endosymbiosis gets engulfed by another eukaryotic cell.
A cell eating a cell that already ate a bacterium.
Exactly. The result is plastids, the general term for chloroplasts and related organelles that have 3 or even 4 membranes surrounding them, remnants of the multiple engulfment events. This explains the origin of chloroplasts and many diverse algal groups like euglenoids, dinoflagellates, and straminopiles. It's crucial for understanding their diversity.
And don't tell me there's tertiary.
Believe it or not, yes. Tertiary endosymbiosis has also occurred. This involves a eukaryotic cell engulfing another eukaryotic cell that contains a plastid derived from secondary endosymbiosis. It's like nested Russian dolls of cells. This has happened in some dinoflagellates. It just highlights the incredibly dynamic and interconnected history of eukaryotic life.
Okay, that incredible story of endosymbiosis fundamentally changed eukaryotes, but it all started with those engulfed bacteria. So let's zoom right in on those foundational life forms, the prokaryotes, and understand why they are still the dominant unseen architects of our world. And maybe tackle viruses, those entities that aren't even really alive in the usual sense, but have such a massive impact.
It's so easy to overlook them, isn't it? Because they're mostly invisible. But prokaryotes, the bacteria and archaea, are, without doubt, the most dominant and successful forms of life on Earth. Their success isn't just about sheer numbers, though those are astronomical. It's about their incredible adaptability and speed. Well, first, their metabolic diversity is just staggering. They can utilize an enormous range of substances for energy and growth. Second, their rapid cell division. Under ideal conditions, a common bacterium like Escherichia coli can double its population every 20 minutes. Imagine that growth rate. And third, their ability to thrive in extreme environments where almost nothing else can survive. We're talking icy Antarctic habitats, the crushing pressures of the deep ocean, near boiling hot springs. The textbook even mentions water near deep-sea vents at 140 degrees Celsius.
140 C, that's well above boiling.
Under pressure, yes. They also survive in environments without free oxygen, using anaerobic processes. Some are actually poisoned by oxygen. And as we established from the molecular data, bacteria and archaea are two very distinct domains of life, as different from each other as either is from us eukaryotes.
So structurally, what makes a prokaryote a prokaryote? What's inside that tiny cell?
The defining feature is the lack of a true nucleus surrounded by a membrane. Their genetic material, usually a single circular DNA molecule, is located in a region called the nucleoid. They might also have plasmid, smaller circular DNA pieces carrying extra genes, maybe for antibiotic resistance or unique metabolic abilities. Almost all have rigid cell walls outside their plasma membrane for protection and shape. They lack the complex internal cytoskeleton of eukaryotes, though they have simpler protein structures involved in maintaining shape and division. They also possess tiny protein-shelled microcompartments containing specific enzymes and often store food reserves like glycogen.
You mentioned cell walls. I remember learning about the Gram stain in biology labs.
Yeah.
It tells you something about the wall, right?
It does. It's a fundamental technique developed by Hans Christian Gram. It differentiates bacteria based on their cell wall structure. Gram-positive bacteria retain the crystal violet dye because they have a relatively thick, single layer of peptidoglycan in their cell wall. Gram-negative bacteria don't retain the dye, they get counterstained pink, because they have a much thinner peptidoglycan layer sandwiched between the inner plasma membrane and an outer membrane made of lipopolysaccharides and proteins. This difference in wall architecture is really significant and affects things like antibiotic susceptibility.
And they can move around and stick to things.
Many can. Motility is often via rotating flagella, which are structurally very different from the flagella of eukaryotes. They don't have microtubules. They also have other filamentous appendages. Fimbriae are typically shorter and more numerous, used for attaching to surfaces or food sources. Pili are often longer and involved in conjugation, a process where genetic material is transferred directly between bacterial cells, like building a temporary bridge. And recently, scientists discovered nanotubes connecting adjacent bacterial cells, forming networks for exchanging molecules even between different species.
So inside they're simple, but they have all these external tools. What about their overall shapes? Are they all just little blobs?
Not at all. While they are simple internally, their shapes are quite diverse and often characteristic. The three major categories are rods, or bacilli, which are cylindrical. Think of Clostridium botulinum, causes botulism, or a bacteria causing fire blight in apples. Cauchi, which are spherical. Examples include Streptococcus lactis, sour's milk, or soil bacteria like Nitrococcus, and spirilla, which are long, curved, or spiral rods. These are less common, but one example is a magnetic bacterium found in swamps. They mainly reproduce by binary fission, simply splitting into two identical daughter cells. Some can also bud or fragment, and some, like Clostridium, can form highly resistant endospores to survive harsh conditions like heat or drying.
Okay, so they're diverse, adaptable, fast reproducing. How does all that translate into their importance for the planet?
Their ecosystem roles are absolutely critical. Without prokaryotes, life as we know it would just collapse. Autotrophic bacteria contribute massively to the global carbon balance through photosynthesis or chemosynthesis. Certain bacteria are unique in their ability to perform nitrogen fixation, converting atmospheric nitrogen gas into ammonia, a form usable by plants and other organisms. This is fundamentally important for all life. Along with fungi, they are the primary decomposers in the biosphere. They break down dead organic matter, recycling essential nutrients back into the ecosystem. Over 90% of the planet's non-human CO2 production comes from these microbial recyclers.
So they're cleaning up and recycling constantly.
Constantly. And this ability is being harnessed for bioremediation. Their capacity to degrade toxic substances, both natural and synthetic like petroleum or pesticides, offers hope for cleaning up pollution. Bacteria are already at work naturally breaking down oil spills, and their commercial and agricultural impacts are huge, producing antibiotics like from actinomycetes, vinegar, amino acids, enzymes. Fermentation by bacteria is essential for making cheese, yogurt, sauerkraut, pickles, and even in biotech. Genetically engineered crops like beet corn use genes from the bacterium Bacillus thuringiensis. There are efforts to re-engineer E. coli to produce biofuels. They are miniature biochemical factories.
Let's dive a bit deeper into the bacteria domain specifically. You mentioned cyanobacteria earlier.
Yes, cyanobacteria are a major group within bacteria. They are the photosynthetic pioneers responsible for originally oxygenating Earth's atmosphere billions of years ago. They come in various forms, unicellular, filamentous like Oscillatoria or nostoc. Some ancient cyanobacteria formed layered structures called stromatolites by binding calcium carbonate. These are abundant in the fossil record and incredibly still form today in some locations. In aquatic environments, many have gas vesicles to regulate their buoyancy, allowing them to float at optimal depths for light. When this regulation fails, they can form massive surface blooms. Some blooms, like those caused by trichodosmium that gave the Red Sea its name, can be harmless, but others secrete toxins dangerous to wildlife and humans.
And they fix nitrogen too.
Many genera do. In the filamentous forms, this often occurs in specialized, thick-walled cells called heterocysts. These heterocysts are adapted for the oxygen-sensitive process of nitrogen fixation. They have thickened walls to limit oxygen diffusion, and they lack the oxygen-producing part of photosynthesis, photosystem 2. They're connected to neighboring cells to exchange nutrients. Cyanobacteria are also common symbionts, living within sponges, protists, fungi and lichens, and even some plants. Some symbiotic ones lose their cell walls and essentially function like chloroplasts within their hosts.
So mostly beneficial, but some bacteria cause plant diseases.
Definitely. Genera like Erwinia, Pseudomonas, Xanthomonas, and Streptomyces causing potato scab are significant plant pathogens. Agrobacterium tumefaciens, which causes crown gall disease, is particularly interesting because it genetically engineers the plant. But then you have crucial mutualists like Rhizobium living in root nodules of legumes, peas, beans, clover, and fixing nitrogen directly for the plant, enriching the soil immensely.
Okay, that covers bacteria. What about the other prokaryotic domain, Archaea? You said they're quite distinct.
Very distinct. Archaea often push the boundaries of where life can exist, and they're turning out to be much more abundant and widespread than originally thought. Physiologically, they are incredibly diverse. The three best studied groups are the extreme halophiles, salt lovers, requiring high salt concentrations, the methanogens, unique in producing methane gas, and the extreme thermophiles, heat lovers. Methanogens are strict anaerobes found in swamps, animal guts, and deep sea vents. They're major contributors to atmospheric methane. Extreme thermophiles thrive at incredibly high temperatures, optimal growth often above 80 degrees C, some even above 110 degrees C. Their enzymes are remarkably heat stable. They often inhabit hot, sulfur-rich environments like Yellowstone's geysers or deep-sea hydrothermal vents. Many metabolize sulfur and are strict anaerobes. Their heat-stable enzymes are very interesting for industrial applications. Importantly, while initially thought to be restricted to these extreme places, Archaea are now known to be major players in less hostile environments like soil and oceans, potentially outnumbering all other organisms in the ocean's plankton. And interestingly, no known pathogens belong to the Archaea.
No pathogens. That's surprising. Okay, so we've covered the prokaryotes. What about viruses? You said they're not quite alive.
Right. They occupy this strange gray area. Viruses are not cells. They lack their own metabolism, ribosomes, the machinery for making proteins. They essentially consist of a genome, either DNA or RNA, single or double-stranded, enclosed in a protein coat called a capsid. Some also have an outer envelope derived from the host cell membrane. They replicate by hijacking the host cell's genetic machinery, forcing it to make more virus components, which then self-assemble into new virus particles.
And they infect everything.
Practically every kind of organism, bacteria, archaea, fungi, protists, plants, animals, they are incredibly diverse and typically very host-specific. We usually discover them because of the diseases they cause. In humans, they're responsible for countless diseases: chickenpox, measles, influenza, colds, hepatitis, polio, rabies, herpes, AIDS, Ebola. The list goes on.
How do they infect plants? Is it different because of the cell wall?
Yes, the rigid plant cell wall presents a barrier. Unlike animal viruses, which often enter cells via specific receptors, plant viruses usually need help getting past a wall. Most commonly, they're transmitted by insect vectors with piercing-sucking mouthparts like aphids or whiteflies that feed on the plant's sap. They can also enter through mechanical wounds, perhaps caused by nematodes feeding on roots, or even during harvesting. Some can be transmitted via infected pollen or spread through vegetative propagation, like planting infected potato tubers.
What do plant viruses look like?
All viruses have protein subunits forming a capsid. Many plant viruses are helical, where the RNA genome fits into grooves of protein subunits arranged in a helix, like the famous tobacco mosaic virus, TMV. Others are icosahedral, forming a 20-sided structure like a geodesic dome, such as many common plant viruses, which are typically very small, around 30 nanometers in diameter.
And once inside, how do they move around the plant?
They have two main routes. For short distances, cell to cell, they move through plasmodesmata, the tiny channels connecting adjacent plant cells. Normally, these channels are too small, but viruses produce special movement proteins that help widen the channels or guide the viral genome through. This is relatively slow, maybe a millimeter a day. For long distance, systemic spread throughout the plant, they get into the phloem sieve tubes, the plant's sugar transport system. This is much faster, maybe a centimeter per day. They travel to growing areas like shoot and root tips, and storage organs like tubers, then re-enter other tissues.
Do plants fight back against viruses?
They do. Plants have sophisticated defense mechanisms. One key strategy is the hypersensitive response, HR. If a plant recognizes a pathogen via specific resistance genes, it can trigger programmed cell death right at the infection site. This sacrifices a few cells, but is often accompanied by the accumulation of antimicrobial molecules, effectively walling off the pathogen and preventing its spread.
Wow. Okay, and viruses aren't even the simplest infectious agents. You mentioned viroids.
Even simpler. Viroids are the minimalists of the infectious world. They are just small, circular, single-stranded RNA molecules. The key distinguishing feature is that they completely lack a protein capsid. They are naked RNA. They're tiny, much smaller than even the smallest viral genomes, maybe only a few hundred nucleotides long. They don't code for any proteins themselves, so they are utterly dependent on the host cell's enzymes, particularly RNA polymerase, to replicate, usually in the host cell's nucleus. They likely cause disease symptoms by interfering with the host's own gene regulation.
Okay, from the absolute minimalists like viroids, let's shift gears to a whole kingdom that's often misunderstood, yet profoundly impacts our world: fungi. I remember learning they used to be grouped with plants, but that's not right, is it?
Not at all. That's a key aha moment from systematics. Fungi are non-motile, typically filamentous eukaryotes. Crucially, they lack plastids and photosynthetic pigments. They can't make their own food like plants. Instead, they are heterotrophs, absorbing nutrients from dead or living organisms. For a long time, yes, they were classified with plants, maybe because they have cell walls and don't move, but molecular comparisons, especially of ribosomal RNA sequences, clearly show they are much more closely related to animals than to plants. They diverged from animal ancestors about 1.5 billion years ago, likely from protists similar to modern nuclearia. So their filamentous growth form is actually convergent with some plant-like protists, not a sign of close relation to true plants.
Okay, more closely related to us than to plants. What are their defining features then?
Key features include cell walls typically containing chitin, the same tough polysaccharide found in insect exoskeletons, not the cellulose of plants. They often form complex structures for producing and dispersing spores, and they have really complex life cycles, often involving both sexual and asexual reproduction.
And that filamentous structure. If you see mold on bread or a mushroom, what are you actually looking at?
We're seeing parts of the fungal body. Most fungi consist of microscopic, thread-like filaments called hyphae. Singular dot hypha. A mass of these hyphae making up the body of a single fungal organism is called a mycelium. Think of the fuzzy mold on food or the extensive white network of a root rot fungus spreading under tree bark, that's the mycelium. These hyphae grow rapidly at their tips, extending the mycelium. A single fungus can produce over a kilometer of new hyphae in just 24 hours.
A kilometer? That's incredible growth. What are they doing with all that growth?
Mostly, they are decomposing or feeding. Fungi are the ultimate recyclers, but that also puts them in direct competition with human interests quite often. They are equipped with incredibly powerful enzymes that can break down complex organic substances, including lignin and cellulose, the main components of wood. Very few other organisms can break down lignin effectively, and they aren't picky. A rotting log in the forest, a wooden fence post, your house timbers, it's all potential food to a fungus.
So they break down tough stuff. What else do they attack?
Pretty much anything organic. The text mentions cloth, paint, leather, waxes, jet fuel, petroleum, paper, insulation, photographic film, even the coatings on CDs and DVDs. They are a major cause of food spoilage, growing on bread, fruits, vegetables, meats, reducing nutritional value and making food unpalatable. Think of rhizopus mold on strawberries. And some fungi produce dangerous mycotoxins when growing on food sources, which can be very harmful as ingested. They're also highly adaptable, growing across wide temperature ranges. Some can grow below freezing, others thrive above 50 degrees C.
Can they cause diseases too, right? Not just spoilage.
Oh, absolutely. Fungi are the most important causal agents of plant diseases. Thousands of species attack crops, garden plants, trees, wild plants. Think of anthracnose diseases causing black lesions, dogwood blight, brown rot of peaches and plums, Panama disease wiping out banana varieties, devastating chestnut blight, Dutch elm disease. The lot is long and economically significant.
You mentioned the Irish potato famine fungus earlier.
Oh,
That was a protist, not a true fungus.
Correct. Phytophthora infestans, late blight, and other water molds like Pythium are umycetes, now classified with protists, stramonopiles, despite their fungus-like appearance and impact. But true fungi cause countless other plant diseases, and fungi also cause animal and human diseases. Over 175 species are known to cause serious infections. This is an increasing concern globally, partly due to growing populations with compromised immune systems, perhaps from AIDS or chemotherapy treatments. Examples include pneumocystis pneumonia, a major killer in AIDS patients, once thought to be a protozoan, now known as a fungus, candida infections, thrush, and cryptococcosis caused by a basidiomycetes fungus.
Okay, so decomposers, major pest.
Yeah.
But they must have some positive uses too. You've mentioned penicillin.
Definitely. Their biochemical abilities are also harnessed for good. Yeasts are prime examples. Vintners use them to produce ethanol for wine, bakers use their carbon dioxide production to make bread rise, and brewers use both for beer. Saccharomyces cerevisiae is the workhorse for most wine, cider, sake, and bread. Different strains are used for lager beers. These strains have been domesticated and improved over centuries. Certain penicillium species are essential for making blue cheeses like Roquefort, Stilton, and Gorgonzola, providing their distinctive flavors and aromas during ripening. And yes, antibiotics. Fleming's discovery of penicillin from penicillium mold revolutionized medicine, saving millions of lives by curing bacterial infections. It was a turning point in human history.
Beyond these direct uses, do fungi interact with other organisms in beneficial ways?
Immensely so. Fungi form some of the most critical symbiotic partnerships in nature, profoundly shaping entire ecosystems. The most widespread and ecologically vital are mycorrhizas. These are mutualistic associations between plant roots and fungi.
Mycorrhiza, fungus root.
Exactly. At least 90% of all vascular plant species form these partnerships. It's almost the rule rather than the exception. These associations are critical for plant nutrition. The fungal hyphae extend far out into the soil, acting like an extension of the plant's root system, vastly increasing the surface area for absorbing water and essential mineral nutrients, especially phosphorus, but also zinc, copper, and manganese. The fungi can also provide protection against root pathogens, like other harmful fungi or nematodes. In return, the fungus gets a steady supply of carbohydrates, sugars, and vitamins produced by the plant through photosynthesis.
So it's a win-win.
A classic win-win. And these underground fungal networks, the mycelia of mycorrhizal fungi, can actually connect multiple plants together, even plants of different species. This network allows for the transfer of water, nutrients, and possibly even signaling molecules between plants. These connections are so vital, especially in forests, that tree seedlings grown in sterile soil often fail to thrive or even die when planted out unless they are inoculated with their specific mycorrhizal fungi. They just can't get enough nutrients on their own.
That's amazing. It's like an underground communication and resource sharing network. Are there different kinds of mycorrhizas?
Yes, broadly, two main types described in the text. Vesicular arbuscular, VA, mycorrhizas, now often called arbuscular mycorrhizas, AM, are the most common. Here, the fungal hyphae actually penetrate the root cells, forming highly branched structures called arbuscules inside the cell. These are the primary sites of nutrient exchange. They might also form storage structures called vesicles. The hyphae still extend out to the soil. The other main type is ectomycorrhizas. Here, the fungal hyphae surround the root cells but don't typically penetrate them. They form a dense sheath around the root tip and a network called the Hardig net that grows between the outer root cells. These are very common on forest trees like pines, oaks, and birches.
Okay, mycorrhizas are partnerships with roots. What about lichens? You mentioned them earlier. That's a fungus plus?
A fungus plus a photosynthetic partner, either green algae or cyanobacteria. So if you've seen those crusty, leafy, or shrubby growths on rocks, tree bark, or soil, especially in harsh environments, that's likely a lichen. It's a composite organism, a tight symbiotic partnership. The fungal partner, the mycobiont, usually an ascomycete fungus, provides the structure, protection, and absorbs water and minerals. The algal or cyanobacterial partner, the photobiont, performs photosynthesis, providing food for both partners.
So they can live where neither could survive alone.
Precisely. Lichens are pioneers, colonizing bare rock, harsh Arctic tundra, or desert soils. The textbook mentions over 350 species in Antarctica, where only 2 vascular plants survive. Some lichens grow within 4 degrees of the South Pole. Lichens with cyanobacteria are also important nitrogen fixers, contributing significant amounts of nitrogen to ecosystems, especially in places like old-growth forests, deserts, and tundra. They are also excellent bioindicators of air quality. Because they absorb nutrients and pollutants directly from the atmosphere and can't easily excrete them, they are very sensitive to toxins like sulfur dioxide. Their health or absence can tell us a lot about air pollution levels.
So fungi are decomposers, pathogens, food sources, medicine makers, and essential symbiotic partners. Quite a range. The textbook mentions some major groups.
Yes, it gives a brief overview. Microsporidia are obligate intracellular animal parasites. Zygomycetes include common molds like rhizopus that rot fruit and some insect parasites. Glomeromycetes are ecologically vital as they are the fungi that form those arbuscular mycorrhizas, the VA type. Ascomycetes are the largest group, the sac fungi, including most yeasts, molds like penicillium, edible morels, and truffles, but also many serious plant pathogens like those causing powdery mildews and chestnut blight. Basidiomycetes are the club fungi, including mushrooms, puffballs, shelf fungi, but also damaging plant pathogens like rusts and smuts. The book details the complex life cycle of black stem rust, Puccinia graminis, which requires 2 hosts, barberry and wheat, to complete its cycle.
It really highlights their critical, often hidden roles, doesn't it? Shaping ecosystems, affecting our food, our health.
Absolutely. Their influence is undeniable, and understanding them is key to managing our world.
Okay, if fungi were a bit of a catch-all before modern classification, protists sound like the ultimate biological catch-all group. Basically all the eukaryotes that aren't animals, plants, or fungi. That must mean incredible diversity.
That's exactly right. As we touched on earlier, Proteus is not a true kingdom reflecting a single evolutionary lineage. It's what we call a paraphyletic group. It includes this hugely heterogeneous collection of organisms, unicellular, colonial, simple, multicellular eukaryotes that just don't fit neatly into the other 3 eukaryotic kingdoms. So yes, it encompasses everything from animal-like heterotrophs, often called protozoa, to plant-like autotrophs, the algae, and even groups like water molds and slime molds that were once considered fungi. The diversity is immense.
Let's focus on the algae within that group. We often picture trees when thinking about photosynthesis, but you mentioned algae, especially marine phytoplankton, are really important for the planet's health.
Hugely important, especially for global carbon cycling. Our planet is mostly water, and algae are abundant in those waters. Through photosynthesis, they take vast amounts of atmospheric carbon dioxide, a major greenhouse gas, and convert it into carbohydrates. Some also incorporate carbon into calcium carbonate shells. A significant portion of this carbon, locked up in algal biomass or shells, eventually sinks to the ocean floor when the algae die, effectively sequestering carbon in the deep ocean. Current estimates suggest marine phytoplankton absorb about half of all the CO2 released from human activities like burning fossil fuels. Understanding how much of that carbon stays locked away long-term is a major area of research.
Half. That's staggering. And their food, too, right? Base of the food web.
Absolutely. Marine algae like haptophytes, diatoms, and dinoflagellates are critical primary producers, forming the base of most aquatic food webs. And think bigger scale, too. Massive brown algae, the kelps, like the giant kelp Macrocystis pyrifera off the California coast, form vast underwater forests. These kelp forests provide crucial habitat, shelter, and food for incredible diversity of fish, invertebrates, sea otters. Some kelps can grow over 60 meters long incredibly quickly, and humans harvest seaweeds too. Kelp is eaten as kombu in East Asia. Red algae like porphyra become nori used for wrapping sushi. They provide minerals, though aren't super high in calories for us.
But sometimes too much algae is a bad thing. You mentioned blooms earlier.
Yes, the dark side of algal productivity. When ecosystems are disturbed, often by human activity like releasing excess nutrients from sewage or agricultural runoff, algae can be freed from their natural limitations. This can lead to population explosions called algal blooms. Sometimes these are so dense they color the water, leading to terms like red tides or brown tides, depending on the pigments in the algae. The problem is, many species that form these blooms produce potent toxins, possibly as a defense against being eaten. These toxins can cause severe illness or death in humans who eat contaminated shellfish, and they lead to massive die-offs of fish, marine mammals, and seabirds.
Are these becoming more common?
Unfortunately, yes. The frequency and severity of harmful algal blooms seem to have increased globally in recent decades. This is strongly linked to declining water quality worldwide and increased nutrient pollution associated with growing human populations and agricultural intensification. It's a serious environmental problem.
But there might be positive applications too, using algae.
There's potential. For example, algal turf scrubbers are being used in some places to improve water quality. Algae are grown intentionally to take up excess nutrients from wastewater or agricultural runoff, cleaning the water and restoring oxygen levels. And the biomass produced by these algae could potentially be harvested and converted into biofuels. Developing cost-effective methods for this is an active area of research.
Okay, let's touch on some of the specific protist groups mentioned. You brought up cristomonads as evidence for secondary endosymbiosis.
Yes, they're a key example. Their chloroplasts have 4 surrounding membranes, and they contain a unique mix of pigments, chlorophylls A and C, carotenoids, and phycobilins, which are otherwise mainly known from cyanobacteria and red algae. This strongly suggests their chloroplasts originated from engulfing another eukaryotic alga that already had chloroplasts derived from a cyanobacterium.
What about haptophytes?
They are very important components of marine plankton, contributing significantly to food webs and also to global cycles. They play a major role in transporting organic carbon and calcium carbonate to the deep ocean. Interestingly, they are also major producers of certain sulfur compounds that contribute to atmospheric sulfur levels and potentially influence cloud formation and climate. One genus, Phaeocystis, can form massive blooms in polar regions.
And dinoflagellates. They cause some red tides, but also have symbiotic forms.
Correct. Many dinoflagellates are photosynthetic, again showing evidence of secondary endosymbiosis in their pigment composition. But they are also famous for the symbiotic forms called zooxanthellae. These are typically golden spherical cells living within the tissues of other marine organisms, most famously corals. These zooxanthellae are the powerhouses of coral reefs. Their photosynthesis provides the vast majority of the energy that allows corals to build their massive calcium carbonate skeletons in nutrient-poor tropical waters. A single cubic millimeter of coral tissue can contain up to 30,000 of these tiny algal cells.
So the core depends on the algae.
Absolutely. The coral provides shelter and nutrients like nitrogen and phosphorus to the algae and stimulates the algae to release most of the carbohydrates they produce, often as glycerol, which the coral uses for its own respiration and growth. This symbiosis is why reef-building corals are restricted to shallow, clear waters where their algal partners get enough light for photosynthesis. Coral shapes are often adapted to maximize light capture.
And this is where coral bleaching comes in.
Exactly. Coral bleaching occurs when the coral expels its zooxanthellae, usually due to environmental stress. The primary trigger is elevated water temperature. Even just a 1–2 degree rise sustained for a few weeks can cause it. Ocean acidification from excess CO2 dissolving in seawater is another major stressor. Without their algae, the corals lose their main source of energy and their color, revealing the white skeleton underneath, hence bleaching. If the stress persists, the coral polyps often die, leading to the degradation of the entire reef ecosystem. It's a stark indicator of climate change impacts.
Moving on, what are streamenophiles?
This is another major eukaryotic supergroup. A key feature, at least ancestrally, is having 2 different types of flagella on motile cells, one smooth and one hairy. This group includes some very important photosynthetic lineages. Like the diatoms, These are incredibly abundant unicellular algae, famous for their intricate cell walls made of silica, essentially glass houses.
Glass houses, you mean their shells?
Yes, their two-part cell wall is called a fristule. It's made of polymerized silica, like opal. The two halves fit together perfectly, like a petri dish. Under an electron microscope, you can see these frustules are covered in incredibly detailed patterns of pores and ridges unique to each species. These patterns are used for identification. When diatoms reproduce asexually, they divide, and each daughter cell gets one half of the parent fristule and makes a new, slightly smaller half that fits inside. This means the average cell size in the population gradually decreases over generations.
They get smaller and smaller. What happens then?
Eventually, when they reach a critically small size, or due to environmental cues, they switch to sexual reproduction. This usually involves forming gametes, fertilization happens, and the resulting zygote grows into a large oncospore, which then forms a new, full-size frustule, restoring the maximum size for the species. The empty silica frustules of dead diatoms sink and accumulate on the ocean floor, forming deep layers over geological time. When these layers are mined, we get diatomaceous earth used in filters, abrasives, and other applications.
What other stratigraphies are important?
Golden algae, Cruciferaceae, are another group often found in freshwater. Brown algae, Phaeophyceae, include the largest protists, the kelps, forming those underwater forests we talked about. Brown algae have cell walls containing algin, a gelatinous substance that provides flexibility against wave action and prevents drying out. Algin is commercially harvested as a stabilizer and emulsifier in things like ice cream, cosmetics, and paints.
Then there are red algae, Rhodophyta. What's unique about them?
They have complex cell walls containing cellulose plus gelatinous polymers called agar and carrageenans. These are widely used commercially as gelling agents and thickeners in foods, like jelly, dairy products, and laboratory media. Some red algae, the coralline algae, deposit calcium carbonate in their walls, becoming hard and stony. They play a crucial role in cementing coral reefs together and can live at remarkable depths over 260 meters down, where light levels are incredibly low. Unfortunately, a bacterial disease is now threatening these important reef builders in the South Pacific. Red algae also have particularly complex life cycles, often involving 3 distinct multicellular generations, which is quite unusual.
And finally, green algae, Chlorophyta. You mentioned these are closely related to land plants.
Yes, this is a really important connection. Green algae and land plants, bryophytes and vascular plants, share several key features that indicate they form a single evolutionary lineage, sometimes called viridiplantae, literally green plants. Both groups contain the same photosynthetic pigments, chlorophyll A and chlorophyll B. Both store their food reserves as starch inside their plastids. Many have cell walls containing cellulose. And the microscopic structure of flagellated cells in some green algae closely resembles the sperm of land plants. Molecular data strongly confirms this close relationship.
Are there specific groups of green algae that are more closely related than others?
Yes. The class Carefischii, in particular, is considered the closest living relative to the ancestor of all land plants, bryophytes, and vascular plants. They share specific details in cell division, like the formation of a phragmoplast and cell plate, and the structure associated with their flagella that are also found in land plants, but not typically in other green algae. These shared derived traits are strong evidence pointing to them as the sister group to land plants.
Okay, so that covers the algae side of protists. What about the heterotrophic ones, the ones maybe previously called fungus-like?
Right. The main groups discussed here are the Umycetes and the slime molds. Umycetes, phylum Umicota, as we mentioned, are the water molds. Despite filamentous growth and causing plant diseases like fungi, their cell walls are mostly cellulose, not chitin, and molecular data places them firmly within the straminopiles related to diatoms and brown algae. They include devastating plant pathogens like Plasma parviticola, downy mildew of grapes, which led to the invention of the first fungicide, Bordeaux mixture, and the genus Phytophthora, plant destroyer. Phytophthora species cause diseases like potato late blight, pea infestans, sudden oak death, P. ramorum, and root rot in countless plants, including avocados, P. cinnamomum. They cause billions of dollars in damage annually. Slime molds are also heterotrophic protists, once considered fungi. The textbook distinguishes between plasmodial slime molds forming large multinucleate masses called plasmodia and cellular slime molds, existing as individual amoeboid cells that aggregate, noting they are unrelated but often discussed together. They are fascinating examples of organisms that blur the lines between unicellular and multicellular life.
All right, we've explored the foundation's classification, prokaryotes, fungi, protists. Now let's turn to a really significant evolutionary leap in plant history. the move onto land, and the development of vascular tissues. This gave rise to the seedless vascular plants.
Yes, this transition from aquatic carefish and green algae ancestors to terrestrial life was monumental. Developing a vascular system specialized tissues for transporting water, xylem, and sugars, phloem, was a complete game changer. It allowed plants to grow much taller, competing for sunlight, and to transport water from the soil up to aerial parts much more efficiently than the simpler non-vascular plants like mosses, bryophytes.
So vascular plants share ancestors with those carefish algae. How do they relate to bryophytes, the mosses, and liverworts?
They both evolved from those algal ancestors and share features like an alternation of generations where the sporophyte and gametophite look different, heteromorphic. But in bradophytes, the gametophite, the haploid generation, is typically the larger, dominant, free-living stage. The sporophyte, diploid generation, remains small and nutritionally dependent on the gametophyte. Bryophytes also need external water for their swimming sperm to reach the egg. They are often called the amphibians of the plant kingdom because of this dependence on water for reproduction. The key innovation in vascular plants was the reversal of dominance. The sporophyte generation became the larger, complex, dominant, free-living stage, while the gametophyte became much reduced. And of course, the development of true vascular tissues, xylem and phloem, was central.
How do these vascular plants grow?
They exhibit primary growth, which is growth originating from apical meristems located at the very tips of roots and stems. This is primarily responsible for increasing the length of the plant body. The tissues produced by this are called primary tissues, making up the primary plant body. Many ancient vascular plants, and even some modern ones like ferns, consist entirely of primary tissues. Later, some lineages evolve secondary growth, which increases the thickness or girth of stems and roots, producing wood. We'll see more of that with seed plants.
You mentioned the stele earlier as the internal plumbing. Can you elaborate on that? How did it change?
The stele is the central cylinder of vascular tissue, xylem and phloem, within the stem or root. Its arrangement provides clues about evolutionary history. The simplest and most ancient type is the protostele. Imagine a solid core of xylem in the center, surrounded by a layer of phloem. This is found in the stems of lycophytes, like club mosses, the roots of most vascular plants, and the stems of many extinct early land plants. A more complex type is the siphonoustele, which has a central core of non-vascular tissue, pith, surrounded by the vascular cylinder. The phloem might be only outside the xylem or on both sides. This is common in the stems of ferns. In ferns, when vascular tissue branches off to supply a leaf trace, it often leaves a break or leaf gap in the vascular cylinder above it. Then there's the eustele, characteristic of almost all seed plants, gymnosperms and angiosperms. Here, the primary vascular tissues are arranged in discrete strands or bundles, typically in a ring surrounding a central pith.
So protostele, siphonoustele, eustele is the linear progression. Did one evolve from the other?
It seems Eucellus, in seed plants, likely evolved directly from Protostellus. Siphonosteles, in ferns and relatives, probably represent a separate evolutionary line, also originating from Protostellus. This is important. It suggests that the lineage leading to modern seed plants diverged quite early, and none of the living groups of seedless vascular plants, like ferns or lycophytes, are direct ancestors of seed plants. They are more like evolutionary cousins that took different paths after branching off from common ancestors.
What about leaves? Did they just appear fully formed?
Not likely. Early vascular plants might have had simple, scale-like outgrowths called innations, which initially lacked vascular tissue. True leaves seem to have evolved along two main lines. Microphyls are thought to have developed when a single, unbranched strand of vascular tissue extended into one of these innations. They are typically small, simple leaves with a single vein and are characteristic of lycophytes, club mosses, spike mosses. They're associated with stems having protosteles. Megaphils, on the other hand, are generally larger leaves with a complex branching system of veins. They are thought to have evolved from entire branch systems that flattened and developed webbing, parenchyma tissue, between the branches. Megaphils are associated with stems having siphonosteles or eusteles, and their departure from the stem creates those leaf gaps in ferns or similar structures, leaf trace gaps in seed plants.
So two different ways leaves evolved. What about reproduction? You mentioned homospore and heterospore.
Right, this relates to the spores produced by the sporophyte generation. Homosporous plants produce only one type of spore. When this spore germinates, it typically grows into a bisexual gamophyte, producing both sperm and eggs, although environmental factors can sometimes influence sex expression. Most living ferns, except water ferns and horsetails, equisetum, are homosporous. Heterosporous plants produce two distinct types of spores in different sporangia. Smaller microspores grow into male gamophytes, producing only sperm, and larger megaspores grow into female gamophytes, producing only eggs. Heterospore evolved independently in several groups, including some lycophytes, Selaginella, Isootes, water ferns, and crucially, it's a prerequisite for the evolution of seeds. All seed plants are heterosporous.
And the structures that make spores, the sporangia, they differ too.
Yes, there's a distinction based on development. Eusporangia develop from multiple initial cells and have walls that are several layers thick. This type is found in lycophytes, horsetails, and some fern groups, like Psilotopsida, Maradiopsida. Leptosporangia develop from a single initial cell and have a wall consisting of just a single layer of cells. This is a more derived feature found only in the largest group of ferns, the Polypodiopsida, which includes most familiar ferns and the water ferns.
Okay, let's walk through the actual groups of seedless vascular plants. The textbook mentions extinct ones first.
Yes, it highlights some key extinct Devonian phyla, Devonian period, roughly 419-359 million years ago, that show early stages of vascular plant evolution. Rhinophyta, like Cooksonia and Rhinia, were small, simple leafless plants with branching stems bearing sporangia at the tips. Rhinia had a simple protostele. Zostera and Trimerophytophyta were other early groups, showing increasing complexity in branching and sporangial arrangement. Then there were the progymnosperms, which we should revisit briefly.
Right, the precursors to seed plants.
Exactly. They appeared later in the Devonian and had characteristics bridging the gap between seedless plants and seed plants. While they reproduced via freely dispersed spores like ferns, they uniquely possessed secondary growth, producing true wood, secondary xylem, and secondary phloem from a vascular cambium, just like modern conifers. Archaeopteris is a key example. It looked like a tree with fern-like leaves, actually flattened branch systems, and had a eustele like seed plants. Some species were even heterosporous. This shows that wood production and heterospory evolved before the seed itself, setting the stage.
Fascinating. Now, what about the seedless vascular plants still living today?
There are two major living phyla. First, phylum Lycopodiophyta, the lycophytes. This includes club mosses, spike mosses, and coolworts. The Lycopodiaceae are the club mosses, like Lycopodium. Many are tropical epiphytes growing on other plants, while temperate ones often creep along the ground forming mats. Their sporophytes have branching stems, rhizomes, roots, and characteristic microphyls, usually arranged spirally. They are homosporous. The sporangia are borne on specialized leaves called sporophylls, which are sometimes clustered into cone-like structures called strobili.
And spike mosses. They're different.
Yes, Selaginella, spike mosses, and Isoetes, coolworts, are distinct from club mosses mainly because they are heterosporous. Selaginella produces microspores and megaspores, usually in the same strobilus. The megaspore develops into a small female gamophyte, mostly within the spore wall, nourished by stored food. The microspore develops into a tiny male gamophyte. Fertilization still requires external water for the sperm to swim to the egg, often happening after the spores have been shed. Selaginella includes the famous resurrection plant, S. lepidophila, that can survive extreme drying.
Okay, lycophytes. What's the other major living phylum?
Phylum Manilophyta. This is a relatively recent grouping based on molecular and morphological data, bringing together ferns, horsetails, and whisk ferns. The vast majority of species in this phylum are ferns. Most familiar ferns belong to the class Polypodiopsida, the leptosporangiate ferns. They are distinguished by having those leptosporangia that develop from a single cell. Most are homosporous, except for the aquatic water ferns, which are heterosporous. There are also eusporangiate ferns in other classes, Psilotopsida, Maradiopsida. Psilotopsida includes whisk ferns, Psilotum, which lack true roots and leaves, looking very primitive, and adder's tongues grape ferns, Ophioglossum botricium, which typically produce a single complex leaf each year divided into a vegetative and a fertile spore-bearing part.
And horsetails are in this group too, Manilophyta.
Yes, horsetails, equisodophytes, are now placed within Manilophyta. There's only one living genus, Equisatum, with about 15 species. It's a remarkable living fossil, almost unchanged from ancestors like equisetites found in 300-million-year-old rocks. Equisatum has distinct jointed stems, often with whorls of reduced scale-like leaves at the nodes. The stems are photosynthetic and contain silica, giving them a rough texture, hence the common name scouring rush. They are homosporous. The sporangia are borne on unique umbrella-shaped structures called sporangiophores, which are clustered into strobili, cones, at the tips of fertile stems. Their spores have unique appendages called elaters that coil and uncoil with changes in humidity, aiding in wind dispersal. The spores germinate into small, free-living endophytes.
Okay, from those fascinating seedless plants relying on spores and often water for reproduction, we move to another monumental evolutionary leap, the seed. This invention really changed the game, didn't it? Allowing plants to conquer drier lands. These are the gymnosperms.
Absolutely. The seed was a revolutionary adaptation for terrestrial life. It's essentially a protected package containing the embryo and a food supply, allowing the new generation to wait for favorable conditions before germinating. It's a key factor in the success of gymnosperms and later angiosperms.
Gymnosperm means naked seed.
That's right. It refers to the fact that their ovules, which develop into seeds after fertilization, are not enclosed within an ovary, part of a flower's carpel. They are typically born exposed, often on the surfaces of scales, like in a pine cone.
So what's needed to make a seed? You mentioned heterospermy earlier.
Yes, heterospermy is a prerequisite. All seed plants produce microspores, leading to male gametophytes, i.e. pollen, and megaspores, leading to female gametophytes. The key innovation is the ovule. An ovule consists of the megasporangium, also called the nucellus, which contains the megaspore, surrounded by one or more protective layers called integuments. There's usually a small opening, the microbial, allowing access for pollen. Crucially, in seed plants, the single functional megaspore is retained within the megasporangium. It germinates inside to form the female gametophyte, which also remains inside the ovule, nourished by the surrounding sporophyte tissue. After fertilization, which also happens inside the ovule, the whole structure integuments, remnants of the nucellus, the female gametophyte, often serving as stored food, and the developing embryo, the new sporophyte, matures into the seed. The integuments become the protective seed coat.
So the whole female gametophyte generation is protected inside the parent plant.
Essentially, yes, until the seed is dispersed. This provides much greater protection and nourishment compared to the free-living gametophytes of most seedless plants.
And gymnosperm ovules differ slightly from flowering plant ovules.
Typically, yes. Gymnosperms usually have only a single integument surrounding the ovule, whereas most angiosperms have two.
We talked about progymnosperms bridging the gap, producing wood and heterospermy before seeds evolved.
Exactly. Groups like Archaeopteris showed that key features of seed plants, like secondary growth, wood, and the vascular pattern, appeared before the ovule and seed fully evolved. They set the evolutionary stage.
Okay, so which gymnosperms are still around today? The textbook discusses 5 phyla of living seed plants overall, right? 4 gymnosperm, 1 angiosperm.
Correct. Seed plants first appeared at least 365 million years ago. Today, there are 4 recognized living phyla of gymnosperms. The most familiar is phylum Coniferophyta, the conifers.
Pines, firs, redwoods, the dominant trees in many forests.
That's them. They are hugely important ecologically and economically. Think timber, paper pulp, resins, turpentine, and habitats for wildlife. The text mentions the whitebark pine, Pinus albicaulis, as vital food for bears and birds, but also threatened by disease and climate change, highlighting their ecological roles and vulnerability.
What's their reproduction like? Pine cones are the key, right?
Yes. Conifers have separate male pollen cones and female ovulate or seed cones. Pollen cones are usually small and non-woody, producing vast amounts of pollen containing the male gametophytes. Ovulate cones are typically larger and become woody. They consist of scales, and on the surface of each scale are usually 2 ovules. Conifers are mostly wind-pollinated. Pollen grains land near the micropyle of an ovule. A pollen tube then grows through the nucellus towards the female gametophyte inside, carrying the sperm nuclei. Fertilization occurs when one sperm nucleus fuses with the egg nucleus inside the female gametophyte. In pines, the other sperm nucleus degenerates. The fertilized ovule then develops into the seed containing the embryo, nu2 and sporophyte, the remains of the female gametophyte, and serving as food reserves, all wrapped in the seed coat derived from the integument, parental two-end tissue. The seed is then dispersed, often when the cone's scales open.
Are all conifers pines and firs?
Oh no, there's significant diversity. The Archaeaceae family, for instance, is found mainly in the southern hemisphere today. It includes the fascinating Willemia nobilis, the Willemi pine, discovered in Australia in 1994, one of the world's rarest plants, a true living fossil. Also includes monkey puzzle trees and Norfolk Island pines, often seen in cultivation. The Cupressaceae family includes cypresses, junipers, and the giants redwoods, sequoia, and giant sequoias, sequoiadendron. Also the deciduous bald cypress, taxodium, and the living fossil dawn redwood, metasequoia. Junipers have distinct fleshy cones that look like berries and are used to flavor gin. The Taxaceae family are the yews. They are unusual among conifers because their single seeds are surrounded by a fleshy, often red, cup-like structure called an aril, which attracts birds for dispersal. But beware, yew seeds and leaves are highly toxic, and the Pinaceae family itself is huge, including pines, pinus, firs, abes, larches, larix, which are deciduous conifers, spruces, picea, and hemlocks, sugo.
Okay, conifers are one phylum. What else? Cycads.
Yes, phylum Cycadophyta, the cycads. These often look like palm trees or large ferns, with a stout trunk and a crown of large, compound leaves. They are ancient plants, often called living fossils, more abundant during the age of dinosaurs. They exhibit true secondary growth, but it's often slow. Many cycads are highly toxic. They also have unique coralloid roots near the soil surface that host nitrogen-fixing cyanobacteria, Anabaena, providing nutrients. Interestingly, while historically thought wind-pollinated, insects, particularly beetles, seem to play a significant role in the pollination of many cycad species.
Another living fossil group.
Indeed. And then there's phylum Ginkgophyta, which has only one living species, Ginkgo biloba. It's instantly recognizable by its unique fan-shaped leaves. Fossils show this lineage dates back about 270 million years. It likely only survived extinction because it was cultivated for centuries in temple gardens in China and Japan. Now, it's widely planted in cities worldwide because it's remarkably tolerant of air pollution. Ginkgo reproduction is fascinating too. Like cycads, it retains the ancestral trait of producing motile flagellated sperm that swim the final distance to the egg, even though it uses a pollen tube initially.
Wow, swimming sperm in a seed plant. What's the last gymnosperm phylum?
Phylum Netophyta, the Netophytes. This is a really enigmatic group with only 3 living genera, Ephedra, Natum, and Wallachia. They are particularly interesting because they share several features with flowering plants, angiosperms, which led to past speculation that they might be closely related. These features include vessel elements in their xylem for water transport, common in angiosperms but rare in other gymnosperms, and their cone structures sometimes resembling flower clusters. Natum and Wallachia even lack archegonia, the structure containing the egg in other gymnosperms and seedless plants, similar to angiosperms.
So are they the closest relatives to flowering plants?
Current molecular evidence suggests probably not. It seems more likely that netophytes are sister to the conifers, or perhaps part of a larger group with conifers and ginkgo. Those angiosperm-like features are now generally thought to have evolved convergently independently in netophytes and angiosperms. Ephedra is also interesting because it exhibits a form of double fertilization, similar to angiosperms, where 2 sperm nuclei are delivered. However, in Ephedra, the second fertilization results in an extra embryo that aborts, not the nutritive endosperm tissue characteristic of angiosperms. And Wallachia mirabilis is just one of the weirdest plants on Earth. Found only in the Namib Desert, it mostly consists of a huge, woody, underground stem root system, with just 2 enormous strap-like leaves that grow continuously from the base and shred at the ends throughout the plant's potentially 1,000-year-plus lifespan.
Okay, from those ancient lineages of gymnosperms, we finally arrive at the group that dominates our modern landscapes.
Yeah.
The angiosperms, the flowering plants, phylum Anthophyta. These are the plants we see everywhere, right? Trees, shrubs, grasses, flowers, crops.
Absolutely. They make up the vast majority of the visible plant world. From the smallest flowering plant, the tiny duckweed Wolfia, maybe a millimeter across, to the tallest, the towering mountain ash Eucalyptus regnans, reaching over 100 meters. The diversity in form, size, and habitat is simply staggering.
Are they all photosynthetic?
The vast majority are free-living photosynthetic autotrophs, yes, but there are exceptions. Around 3,000 species are parasitic, tapping into host plants for water and nutrients. Mistletoes are a familiar example, as is the non-photosynthetic daughter, Cuscuta, or the bizarre Rafflesia, which produces the world's largest flower but lives mostly inside its host vine. These parasites use specialized structures called haustoria to penetrate host tissues. There are also mycoheterotrophic plants. These lack chlorophyll and don't photosynthesize. Instead, they get their nutrients by parasitizing the mycorrhizal fungi that are connected to other photosynthetic plants. It's an indirect form of parasitism.
Fascinating variation. But the defining feature is the flower, right?
Yes, the flower is the key reproductive structure and a major reason for angiosperm success. It's essentially a modified shoot with specialized leaves adapted for reproduction. The arrangement of floral parts, especially the ovary position relative to the attachment of sepals, petals, and stamens, is an important feature used in classification and understanding evolutionary trends.
Can you explain those ovary positions, hypogynous, progynous?
Sure. In a hypogynous flower, the sepals, petals, and stamens are attached to the receptacle below the ovary. The ovary is said to be superior. Think of a buttercup or a tomato flower. In a pyrogenous flower, the sepals, petals, and stamens are fused at their bases to form a cup-shaped structure called a hypanthium, which surrounds the ovary but is not fused to it. These parts appear attached around the ovary, which is still superior. This is common in the rose family, like cherries or roses themselves. In an epigynous flower, the hypanthium is fused to the ovary wall, so the sepals, petals, and stamens appear to be attached on top of the ovary. The ovary is said to be inferior. Examples include apple flowers, sunflowers, or carrots. These different arrangements likely offer varying degrees of protection to the developing ovules.
Okay, so the flower structure is key. What about the life cycle? Anything unique there compared to gymnosperms?
The most remarkable and defining feature of the angiosperm life cycle is double fertilization. This is unique to flowering plants, though, as mentioned, a phaedra has something superficially similar but functionally different.
Double fertilization?
Yeah.
What gets fertilized twice?
Remember, the pollen grain delivers two sperm nuclei down the pollen tube to the ovule. In angiosperms, both sperm nuclei are functional and participate in fertilization events. One sperm nucleus fuses with the egg cell N to form the diploid 2N zygote, which develops into the embryo. The other sperm nucleus fuses with the central cell of the female gadophyte, which typically contains two polar nuclei, N plus N. This fusion forms the triploid 3N primary endosperm nucleus. This triploid nucleus then divides to form the endosperm, a specialized nutritive tissue that nourishes the developing embryo.
Ah, so one fertilization makes a baby plant, embryo. The other makes its packed lunch, endosperm.
That's a great analogy. The simultaneous formation of both the embryo and its dedicated food supply within the seed is incredibly efficient and considered a major factor in angiosperm success. In gymnosperms, the food supply is the tissue of the female gametophyte itself, which develops before fertilization occurs, potentially wasting resources if fertilization fails.
Smart strategy. Does pollen itself give clues about angiosperm evolution?
It does. The structure of pollen grains is quite informative. The earliest fossil angiosperms, as well as living basal lineages and the monocots, typically have pollen with a single aperture, a groove or pore. This is called monoaperturate pollen and is considered the ancestral type for angiosperms. However, the vast majority of flowering plants belong to a group called the eudicots. A key derived feature uniting the eudicots is their characteristic triaperturate pollen, or types derived from it, meaning pollen grains with three apertures, usually furrows or pores. This difference in pollen structure is a major distinguishing feature between these huge groups.
So based on features like pollen, flowers, and molecular data, how are angiosperms related to each other?
Molecular phylogenetics has really clarified the picture. We now recognize several major lineages. There are a few early diverging basal angiosperm lineages like water lilies and ambarella. Then the two largest groups are the monocots and the eudicots. Monocots, grasses, lilies, orchids, palms, are clearly a monophyletic group, defined by features like having only one cotyledon, seed leaf, flower parts, typically in threes, parallel leaf venation, and that monoaperturate pollen. Eudicots, most broadleaf plants, roses, sunflowers, oaks, beans, buttercups, are also monophyletic, defined primarily by their triaperturate pollen and usually having two cotyledons, net-like leaf venation, and flower parts in fours or fives. Eudicots are incredibly diverse and are further subdivided into major clades like the rosids and astrids, each containing many familiar plant families. These groups often differ in subtle details of ovule structure or flower chemistry.
And where do they come from? Who are their ancestors?
That's still a bit of a mystery, often called Darwin's abominable mystery. Scientists have looked for links among extinct groups like Mesozoic seed ferns, Catoniales, or the Benectitates, which had flower-like structures. The earliest definitive angiosperm fossil currently known is Archifructus, found in China.
Right, the aquatic one from 125 million years ago.
Exactly. It was herbaceous, lived in water, and had relatively simple reproductive structures without showy petals. The stamens, male parts, and carpels, female parts containing ovules, were arranged along an elongated axis. Its aquatic nature has led some to suggest that early angiosperm evolution might have occurred in wet or aquatic habitats, though that's still debated. It gives us a fascinating glimpse into what the very first flowering plants might have looked like.
Okay, we've established the basics of angiosperms, flowers, double fertilization, the major groups. Now, let's really dive into the incredible diversity within this phylum, especially focusing on the flower itself and its amazing interactions with pollinators.
It's a story of increasing specialization. The flowers of those earliest angiosperms, like Archifrictus, were likely quite variable, maybe with unfixed numbers of parts. But over evolutionary time, particularly as relationships with animal pollinators became more specific and refined, floral patterns tended to become more stereotyped within different lineages. Modern plant families often have very characteristic numbers and arrangements of floral parts.
How did petals and sepals actually evolve? Were they always there?
Not necessarily in their current form. Many petals are thought to have evolved from stamens that became sterilized. They lost their pollen-producing function, and the filament perhaps flattened and became colorful to attract pollinators. Like stamens, petals are usually supplied by just one vascular strand. Sepals, the outermost whorl, usually green and protective, are more leaf-like. They are typically supplied by the same number of vascular strands as a foliage leaf, often three or more, suggesting they likely evolved from modified leaves. Nectaries, the glands producing sugary nectar as a reward for pollinators, seem to have arisen in various ways, not necessarily from modified stamens.
What about the carpels, the female parts containing the ovules?
There's evolution there too. Some early diverging angiosperms have carpels that are still somewhat leaf-like and may not even be fully sealed, though pollen still lands on a receptive surface, not directly on the ovule like in gymnosperms. But in the vast majority of modern angiosperms, carpels are fully closed structures, differentiated into the stigma, receptive tip for pollen, the style, stalk connecting stigma to ovary, and the ovary, base containing the ovules. This closed carpel protects the ovule and allows for more precise control over fertilization.
So increasing specialization seems to be a theme.
Definitely. As angiosperms diversified and co-evolved with animal pollinators, their floral structures became more efficient and specific, leading to the incredible array of flower forms we see today.
You mentioned animal pollinators. This seems like a huge factor in angiosperm evolution, using animals to move pollen around.
It's absolutely central. Plants are rooted, they can't move to find a mate. Flowers, by attracting animals and directing their behavior, effectively give plants directed mobility for their pollen. This allows for efficient cross-pollination, transfer of pollen between different individuals, which increases genetic diversity and adaptability. The intricate relationships between flowers and their pollinators are classic examples of coevolution.
What are some of these co-evolved strategies?
They can be quite elaborate. We mentioned food deception in orchids earlier, mimicking flowers that offer nectar or pollen but providing no actual reward, essentially tricking the pollinator. Even more specialized is sexual deception, also common in orchids. Here, the flower mimics the appearance and, critically, the scent pheromones of a female insect. Male insects are lured in, attempt to mate with the flower, and inadvertently pick up or deposit pollen.
Wow, trickery. What about more straightforward signals like color?
Flower colors are definitely not random. They are highly evolved visual advertisements targeted at specific pollinators. Different pigments create the colors. Carotenoids, also found in carrots and autumn leaves, produce yellows, oranges, and reds. They are oil-soluble pigments located in plastids. Flavonoids are another major group. Within flavonoids, the anthocyanins are water-soluble pigments found in cell vacuoles. They are responsible for most reds, pinks, purples, and blues. The exact color of an anthocyanin can depend on the pH acidity of the cell sap. The same pigment might be red in acidic conditions, violet in neutral, and blue in alkaline. Plants can manipulate cell pH to fine-tune color.
So a blue flower might have the same pigment as a red one, just in a different chemical environment.
It's possible, yes. And some flowers even change color after pollination. They might shift from a conspicuous color to a less attractive one, signaling to pollinators, I'm done, move along, no more reward here. This directs pollinators to unpollinated flowers, increasing efficiency. There's another group of pigments, betalains, including reddish beta-cyanins, found only in one specific order of angiosperms, Caryophyllles, which includes cacti, beets, and bougainvillea. They produce reds and yellows, but are chemically distinct from anthocyanins.
So different pollinators respond to different signals,
Right.
like syndromes.
Exactly. We talk about pollination syndromes, suites of floral traits that have evolved in response to specific types of pollinators. Beetles, possibly the earliest insect pollinators, are often attracted to large, solitary, pale, or whitish flowers with strong, fruity, or spicy odors. Bees are highly efficient pollinators. They are attracted to yellows and blues, often see ultraviolet patterns invisible to us, nectar guides, and need landing platforms. They generally don't see red well. Moths, being active at night, are drawn to pale or white flowers that are strongly fragrant, especially in the evening. Butterflies like bright colors, including red, and often visit flowers with long, narrow tubes accessible to their long proboscis. Some flies are attracted to flowers that mimic rotting flesh, dull red or brownish colors, and foul odors. Birds, especially hummingbirds in the Americas, have high energy needs. Bird-pollinated flowers often produce abundant, watery nectar, are typically bright red or orange, colors birds see well, lack fragrance, or birds often have a poor sense of smell, and lack landing platforms as birds hover. Bats, also with high energy needs and active at night, visit flowers that are often large, dull-colored, produce copious nectar and pollen, may hang down excessively, and emit strong fermenting or musky odors.
So it's a very targeted approach. What if there are no animals around?
Some angiosperms have secondarily evolved wind pollination. This is common in grasses, oaks, birches, ragweed. These plants typically produce enormous amounts of small, lightweight, non-sticky pollen. Their flowers are usually inconspicuous, lacking petals and nectar, and they often have large, feathery stigmas to effectively trap airborne pollen. And a few angiosperms living in water have adapted to water pollination, with specialized mechanisms for pollen release and capture at or below the water surface.
Okay, pollination is step one. After fertilization, the flower develops into a fruit. That's all about seed dispersal, right? Another coevolutionary story.
Absolutely. The fruit is essentially the mature ovary, and sometimes other associated floral parts, that encloses the seeds. Its structure is primarily adapted for seed dispersal. Fruits come in countless forms. Dry fruits might split open, dehiscent, to release seeds, like the legumes of peas and beans, or capsules like poppies. Or they might be indehiscent, where the fruit itself is the unit of dispersal, like the caryopsis grain of grasses, where the fruit wall is fused to the seed coat. Or nuts, like acorns with a hard pericarp. Fleshy fruits, berries, drupes like peaches, pomes like apples, are typically adapted for dispersal by animals.
How do fruits help dispersal?
Various ways. Wind dispersal uses adaptations like wings, maple, ash fruits, or plumes, dandelion, thistle fruit seeds, to catch the wind. Water dispersal is common for plants near water. Fruits like coconuts have buoyant tissues, allowing them to float on ocean currents for long distances. Animal dispersal is incredibly diverse. Fleshy fruits entice animals to eat them. The seeds usually pass unharmed through the digestive tract and are deposited elsewhere, often with a bit of fertilizer. Other fruits have hooks or barbs, like burrs, that cling to animal fur or feathers. A special case is myrmecophore seed dispersal by ants. Some plants produce seeds with a nutritious attachment called an eleosome. Ants collect the seed, carry it back to their nest, eat the eleosome, and discard the unharmed seed, often in a nutrient-rich waste pile perfect for germination. And some plants use explosive dispersal, where the fruit dries and splits open violently, flinging the seeds away, like in touch-me-nots in patients.
It seems like plants have evolved an incredible chemical arsenal, too. Not just for attracting pollinators, but maybe for defense.
Definitely. The relationships between plants and animals involve both attraction and repulsion. Plants produce a vast array of secondary metabolites compounds not directly involved in primary metabolism like growth or photosynthesis. Many of these chemicals function as defenses against herbivores or pathogens. For example, plants can produce proteinase inhibitors when wounded, interfering with insects' digestion. Some produce molecules that mimic insect hormones, disrupting your development. Angiosperms, in particular, have an incredibly diverse repertoire of these secondary compounds compared to other plant groups. This chemical diversity is likely another key factor in their ecological success. And many of these compounds that deter insects or pathogens also happen to have physiological effects on vertebrates, including humans. This is the source of many important medicines, but also spices, perfumes, and poisons. Think of caffeine, nicotine, morphine, quinine, digitalis, heart medication, Taxol, anti-cancer. All secondary metabolites evolved by plants, likely for defense or interaction.
Wow. We've journeyed through this incredible diversity classification. Microbes, fungi, protists, the evolution of land plants, seeds, flowers. Now let's bring it all back home to us, to humanity. Our story seems completely interwoven with the story of plants.
It absolutely is. Our species, Homo sapiens, is a relative newcomer on the geological stage, appearing perhaps 500,000 years ago. But our impact, especially in recent millennia, has been unparalleled. And that impact is largely tied to a fundamental shift in how we interact with the plant world.
Before agriculture, we were hunter-gatherers.
Yes. For most of human history, going back to early Homo species maybe 2 million years ago, we subsisted by gathering edible plant parts, fruits, seeds, nuts, leaves, roots supplemented by scavenging and hunting. Learning to control fire perhaps 1.4 million years ago was a major step allowing cooking and protection. Think of groups like the San people of the Kalahari, whose traditional lifestyle involved intimate knowledge of their plant environment.
But then came agriculture. You called it perhaps the single most important innovation.
Arguably, yes. The shift to deliberate planting and cultivation, agriculture, began independently in several parts of us, plummeted around 13,000 to 11,000 years ago, starting notably in the Fertile Crescent of the Near East. It likely wasn't a sudden invention, but a gradual process. People gathering wild cereals like barley and wheat, which thrive in disturbed areas, might have noticed them growing near campsites from spilled seeds. Deliberately saving and planting seeds would have led to more reliable food sources. Over time, continuous management, selecting the best seeds, watering, weeding, protecting from pests, intensified into cultivation.
And this led to domestication, changing the plants themselves.
Exactly. Domestication involves genetic changes in plant populations brought about by human selection, whether conscious or unconscious, for staple crops like cereals, barley, wheat, rice, maize. Key changes included selection for larger grains, stronger stalks, less shattering, seeds that separate easily from chaff, better flavor, and crucially, the loss of natural seed dispersal mechanisms. Domesticated grains hold onto their ripe seeds, waiting for humans to harvest them. Wild grasses scatter their seeds widely. This created a profound interdependence. Crop plants became reliant on humans for propagation, and human societies became fundamentally reliant on these crops for sustenance.
Did agriculture start just in the Fertile Crescent?
No, it arose independently in multiple centers around the globe, each based on locally available wild plants. Besides the Fertile Crescent, wheat, barley, lentils, peas, another major center was in China, along the Yellow River, where millets and later rice, Oryza sativa, were domesticated. Soybeans, glycine max, also became a crucial crop there. In tropical Asia, rice was also key, along with different legumes, taro, yams, mango, and citrus. In Africa, crops like cowpeas, okra, yams, diascorea, coffee, sorghum, and pearl millet were domesticated along with cotton. And in the New World, before European contact, agriculture developed entirely independently. In Mesoamerica, Mexico, Central America, pumpkins and squashes were early domesticates, followed by the cornerstone crop, maize, zea maize, along with beans, chili peppers, tomatoes, cacao, and cotton. Maize is particularly interesting as it's essentially an agricultural artifact, so modified from its wild ancestor, teosinte, that it cannot survive without human cultivation. In the south-central Andes of South America, a distinct agriculture arose based on tuber crops, especially potatoes, Solanum tuberosum, and related species, as well as seed crops like quinoa.
So agriculture allowed human populations to grow enormously.
Dramatically. It provided a more stable and abundant food supply, allowing for larger, settled populations. Hunter-gatherer numbers were likely limited by factors like the difficulty of carrying infants while nomadic. Agriculture removed many of those constraints. The human population exploded from an estimated 5 million globally 10,500 years ago to over 8 billion today. Humans and their domesticated livestock now constitute something like 97% of the total biomass of terrestrial vertebrates, a staggering shift from less than 1% before agriculture. But this success came with costs. Large herds of grazing animals supported by agriculture led to significant environmental degradation and desertification in many regions, both historically and continuing today, with practices like clearing tropical forests for pasture.
And feeding this huge population presents enormous challenges now, doesn't it? The food security crisis.
It's one of the most pressing global challenges. The population is projected to hit 9 billion by 2050. Currently, over 1 billion people suffer from malnutrition. Our current agricultural systems have limitations. Humans already consume, waste, or divert something like 40% of the total net primary productivity on land. Since 1950, world grain production increased dramatically, but this intensification came at the cost of significant topsoil loss and land degradation. Worryingly, in the first decade of the 21st century, grain yields actually declined in about a third of countries for major crops like wheat, rice, maize, and soybeans. Rising food costs hit the world's poorest hardest, those who may spend 80–90% of their income just to eat. And developing sustainable, high-yield agriculture, especially in the tropics where population growth is highest, remains a major hurdle.
What's being done to address this? I've heard of the Green Revolution.
Yes. The Green Revolution, starting around the mid-20th century, involved intensive efforts to increase crop yields, particularly for grains like wheat and rice. A key figure was Norman Borlaug, who won the Nobel Peace Prize in 1970. He developed new, high-yielding dwarf varieties of wheat that didn't fall over when heavily fertilized. These varieties dramatically boosted production in countries like Mexico, India, and Pakistan. averting widespread famine at the time. More recent initiatives, like the Alliance for a Green Revolution in Africa, AGRA, aim to adapt these principles, focusing on diverse crops suited to local conditions, improving soil health, and ensuring farmers benefit.
But focusing on just a few high-yield varieties, didn't that create new problems? You mentioned genetic uniformity being risky.
Precisely. That's the double-edged sword. While the Green Revolution increased yields, it often relied on widespread planting of genetically uniform varieties. This narrows genetic diversity, making the entire crop highly vulnerable if a new disease or pest emerges to which that variety is susceptible. The southern cornleaf blight epidemic in the U.S. in 1970 is a stark example. A new strain of fungus devastated the corn crop because most of it carried the same genetic susceptibility factor. It caused billions in losses. This highlights the critical importance of preserving genetic diversity. We need to actively collect and conserve different strains of our crops, traditional cultivated varieties, land races, and their wild relatives in seed banks and clonal banks, germplasm collections. These genetic resources are essential reservoirs of genes for disease resistance, tolerance to drought or salt, improved nutritional value, and other traits needed for future crop improvement. The Irish potato famine is the classic historical lesson. Reliance on one susceptible variety led to disaster. Breeding using diverse potato relatives restored the crop.
So we need to look beyond our main crops, too, to wild plants.
Absolutely. We rely heavily on a tiny fraction of the plant kingdom for food. Maybe 6 species provide over 80% of human calories. Only about 150 species have ever been widely cultivated. There's vast untapped potential in thousands of other wild or locally cultivated plants. The textbook highlights jojoba, Simmondsia chinensis, a desert shrub whose seeds contain a liquid wax valuable as an industrial lubricant, replacing sperm whale oil. It's now cultivated in arid regions worldwide. Research into developing salt-tolerant crops is also vital, using plants naturally adapted to saline conditions like fodder shrubs, Atriplex, or breeding tolerance into existing crops. This could open up vast areas of arid land and coastal deserts for agriculture.
Plants aren't just food, though. They're also sources of drugs, spices, beverages.
Incredible biochemical factories. About a quarter of all prescription medications are derived directly from plants or are synthesized based on plant compounds. Plants often produce incredibly complex molecules, like steroids used to make cortisone or hormones for birth control pills that are very difficult or expensive to synthesize chemically. They are an inexhaustible source for discovering new drugs. Ethnobotany, the study of how indigenous peoples use plants, is a crucial tool for finding new medicinal leads. The anti-cancer drugs vinblastine and vincristine, for example, were discovered from the rosy periwinkle, Catharanthus rostius, a plant used in traditional medicine in Madagascar. These drugs dramatically improve survival rates for certain cancers like Hodgkin's disease.
And spices and herbs, they've shaped history too, haven't they?
They certainly have. Spices, typically aromatic roots, bark, seeds, fruits from tropical plants, and herbs, usually leaves, often from temperate plants, have been used for millennia for flavoring, preservation, and medicine. The quest for valuable tropical Asian spices like cinnamon, cloves, nutmeg, and especially black pepper drove European exploration, trade, and conflict for centuries from the 13th century onwards. Nutmeg is interesting. The spice mace comes from the fleshy aril surrounding the seed. Familiar Mediterranean herbs like thyme, mint, basil, oregano, mint family, and parsley, dill, coriander, parsley family, are also derived from specific plant secondary metabolites.
And finally, beverages.
2 caffeine-containing beverages dominate globally. Coffee, made from the roasted seeds, beans of Coffea arabica, native to Africa, and tea from the dried leaves of Camellia sinensis, native to Asia. Both rely on the stimulant caffeine, another plant secondary metabolite. Of course, countless other plants provide essential commercial products: rubber, hevea, oil palm, elias, cacao, theobroma for chocolate, sugar cane and sugar beets for sugar, fibers like cotton. The list is immense. Our reliance on the plant kingdom is total. Hashtag outro.
Wow, what an incredible journey we've just taken. From the precise science of classifying life to the unseen dominance of prokaryotes, the fungal partners in our forests, the tiny yet mighty protists, the evolutionary leaps of seedless vascular plants, the ancient lineage of gymnosperms, and the sheer adaptive brilliance of flowering plants. And then bringing it all home to our own species, Homo sapiens, our profound, intricate, and, well, sometimes challenging relationship with the plant world.
What this deep dive truly reveals, I think, is how every level of biological diversity, from the molecular right up to the ecosystem scale, is just completely interwoven. Understanding these connections isn't just academic curiosity, it's absolutely essential for tackling the global challenges we face: food security, climate change, biodiversity loss, human health. This journey through the diversity section should hopefully give you a real foundational appreciation for the astonishing complexity and beauty of life, and, importantly, our place within it all.
So what does this all mean for you listening in? Perhaps the next time you look at a plant, any plant, in a park, in your garden, even the food on your plate, maybe you'll see it a bit differently. Not just as a static object, but as a dynamic player in billions of years of evolutionary history, connected to vast networks of other organisms and profoundly shaping the world around us. It really makes you wonder, doesn't it, what other secrets this incredible diversity still holds, just waiting to be discovered. Thank you so much for joining us on this deep dive into plant diversity. We hope you feel a little more well-informed and perhaps a lot more curious about the living world all around you.
Indeed. Until next time, keep exploring, keep questioning, and keep appreciating the incredible tapestry of life.